So, special thanks to VWR Aventor for organizing this webinar. Before going into the technical discussion, although Kathy talked about QuantaBio a little bit, but I would like to tell you a little bit more about QuantaBio. QuantaBio as a company was formed in the year 2001 by former amplification experts from Life Technologies. QuantaBio is developing pioneering technologies, as Kathy already mentioned, focusing on critical cloning, PCR, qPCR, next generation sequencing based applications. And many of you know that QuantaBio is best known for its pioneering tough mix chemistry for superior performance and extreme inhibitor tolerance. And we are reaching out to researchers and our customers exclusively via VWR. So we started as Quanta Biosciences Inc., and then the name of the company was changed to Quanta Bio in the year 2012. And we went through an evolution of product line as well. So from cDNA synthesis, qPCR reagents to different kinds of PCR reagents, RT-qPCR reagents, and including recent NGS kits. And I would like to mention here that we are currently developing a RNA sequencing kit, which will be a game changer for the NGS market. So stay tuned. So today I would like to discuss three topics. First, miniaturizing and optimizing DNA library preparation to 384 well formats for high throughput sequencing. Then I'll talk about accelerating speed and performance of 16S workflows, preferably in PCR inhibited samples. And at the end, I'll talk a little bit about how you can use QuantaBio products for better structural variant detection in long-range sequencing. So let's start with the first topic, miniaturizing and optimizing DNA library preparation to 384 OL formats. So before going into the 384 OL format, let's talk about QuantaBio NGS portfolio a little bit. So as you know that Illumina is offering a wide range of sequencing platforms. From left to right, the instruments are generating more and more data. That means higher throughput. And researchers can choose appropriate sequencing platform depending on their sequencing need. For example, if somebody is working on targeted sequencing and they need smaller amount of data or lower amount of data, then they can definitely use something like MiSeq or MiniSeq. But if somebody is working with whole genome sequencing of larger genome, like human genome, which requires larger amounts of data, in that case, NovaSeq will be a better choice in that kind of situation. Illumina-based NGS workflow has 4 distinct steps, starting with input DNA, followed by library preparation. Now, library preparation itself has multiple steps, including DNA fragmentation. If your DNA is not already fragmented, like FFPE or circular free DNA, you have to fragment your genomic DNA to start with. And then next step is adapter ligation. And if needed, library amplification at the end. In the Illumina workflow, next step is sequencing the libraries using any of the Illumina sequencing platform as I showed in the last slide. And finally, analyzing the sequencing data. Data analysis most of the time includes mapping short reads into reference genome. Many different factors influence library qualities. For example, quality of the DNA, bias and consistency during the fragmentation step, and efficiency of the enzymes during end repair, A-tailing, and adapter ligation steps. Also, amplification bias during the PCR amplification step may have some adverse effect on the quality of the sequencing data. We may lose DNA during multiple bit cleanup steps involved in the library preparation and during physical transfer of DNA from one tube to another. Spark Frag and Library Prep Kit from QuantaBio enables an user-friendly and streamlined workflow by simply reducing the number of steps and via high-end formulation. So Spark Frag and Library Prep Kit or workflow has 3 simple steps, starting with DNA frag and polishing. Second step is adapter ligation, and the last step is library amplification. So we enable or we go through the library preparation with 3 very simple steps. I'll go through those steps one by one. Starting with fragmentation. So fragmentation can be done in multiple different ways. It can be done via mechanical shearing like Covaris, or tagmentation, or via enzymatic methods. For enzymatic shearing, it can be done using endonuclease cocktail or non-specific nucleases. Now, it has some advantages. For example, enzymatic shearing is high throughput, cost-effective, and automation-friendly. But it also comes with a risk of sequence bias, lack of reproducibility, etc. Now, I'll show you how Spark, Frag, and Library Prep overcome these challenges of enzymatic shearing. So here you can see that Spark, Frag, and Library Prep Kit shows distinct step down in fragment size with changes in incubation time. So if you increase the incubation time, the fragment size is getting shorter and shorter. And sharply defined fragmentation profile, as you can see from this graph, is closely resembles Covaris shearing. Proprietary frag and polishing enzyme mix ensures a tunable and reproducible fragmentation profile across a range of DNA input. And fragmentation enzymes work on a wide range of DNA input, typically from 1 nanogram to 1 microgram. As you can see from this graph, here we are showing data for 10 nanogram, 100 nanogram, and 1 microgram. It works really well with 1 nanogram DNA as well. As I said, second step is the ligation. A fast ligation process shows an excellent dynamic range and consistent library preparation efficiency over a wide range of DNA input. If you look at the graph here, you will appreciate that PCR-free library yield is in a linear correlation with input DNA. So here on the y-axis, this is the yield of PCR-free library, and in the x-axis, it's input DNA. And this high efficiency ligation was validated with various adapters from different vendors with input DNA from 0.1 nanogram to 1 microgram. Now I'll talk about the last step, which is library amplification step. So when we compared sequence biased between PCR-free library and the libraries that were amplified. So in this graph, the orange one is amplified library and the blue one is PCR-free library. In both cases, we did not observe any bias. And definitely we did not observe any bias or additional bias introduced by hi-fi amplification step. And we also looked into the yield of our library amplification step using Spark Hi-Fi master mix with a few other master mixes available in the market, and we were observing higher yield compared to other competitors in the market. We also looked into the duplication rate between PCR-free and amplified library. In each case, the duplication rate was very, very low, as you can see from this table in the bottom. And definitely, we can say that PCR steps did not increase the presence of duplication rate. So if somebody need to amplify their libraries at the end to enrich the libraries, definitely that's not going to increase the duplication rate. So that is really important. Here we are looking at the libraries that were prepared using Spark Kit and other few kits available in the market. And we are looking at the genomic coverage at wide range of GC content. And if you look at the orange curve here, you will see that libraries prepared using Spark Kit showed equal coverage of the genome having GC content between 10% to 85%. So it works equally well in a genome with higher AT and higher GC regions of the genome. So let's move the discussion towards high throughput library preparation at 384OL format. So first thing is, what are the benefits of library preparation at 384OL format? Why do you want to do that at the first place? So library preparation at 384OL format will not only increase the speed of the library preparation almost 4 times, but it will also significantly reduce the library preparation cost. So it will increase the speed and reduce the cost, so which is great. And most importantly, these benefits will allow diagnostic labs to increase their testing capabilities 4 times compared to their current workflow. But it comes with multiple challenges. There is a speculation that the efficiency of the reactions associated with the library prep may alter at lower reaction volumes, because at 384 workflow, your reaction volumes are going to be much lower. And obviously, bead purification at lower volume is extremely challenging. Just to give you a prospect here, the maximum reaction volume for the library preparation at 96 format are typically 50 to 100 microliter. And during the bead cleanup step, when you are adding beads and doing ethanol wash, the maximum volume goes to 180 to 200 microliter. Now, on the other hand, the maximum volume a 384OL PCR plate can handle is 50 microliter. And remember, you have to dip the tip into it in order to remove or add more volume into it. So the recommended reaction volume is between 1 to 35 microliter. So this is really, really challenging. But In the lab, we were able to optimize library preparation at 384 OL format using Spark, Frag, and Library Prep Kit. And I would like to share some data with you. So here we are showing some example tape station traces of the libraries that were prepared with 384 OL format using 100 nanogram human genomic DNA as input. And you can clearly see here that with the increase of fragmentation time, the size of the library is going down. Similarly, we saw for 96OL format. So we were able to maintain a tunable and reproducible fragmentation profile at 384OL format as well. So similar to 96OL format, depending on what library size you want, you can choose a certain fragmentation time and go from there. Similar workflow. And we were able to achieve 500 to 700 nanogram libraries with 6 cycles of PCR when we used 100 nanogram of human genomic DNA as an input. Although I am showing data for 100 nanogram human genomic DNA here, but I want to tell you that we tested this protocol for 500 nanogram or 50 nanogram DNA and it worked equally well. So this protocol can be adapted at different DNA inputs. So here we compared the quality of the libraries prepared with 384OL format and 96OL format. The blue and orange traces here are the libraries prepared with 384OL format using 100 nanogram DNA as input, and libraries were amplified with 6 cycles of PCR. And the fragmentation times were 24-minute and 20-minute respectively. And here the green trace is from a library that was prepared with 96OL format using same 100 nanogram DNA input, 5 cycles of PCR, and fragmentation time was 16 minutes. And if you look at the tape station traces, you can clearly see that the quality of the libraries prepared with 384 and 96OL formats have similar characteristics. The only difference is that 384OL formats need little longer fragmentation time and 1 extra cycles of PCR in order to achieve same quality and similar yields compared to the 96OL format. So if a lab wants to improve their throughput, wants to move from 96 to 384OL format, they can readily use our protocol and use the Spark, Frag, and Library Prep Kit for their applications. So we have learned so far that Spark, NGS, Frag, and Library Prep Kit enables a smooth, streamlined workflow with minimum steps and superior performance. To reduce the cost of library preparation and increase the throughput, libraries can be prepared with 380OL format. And as I said, the protocol is ready to go with Spark, Frag, and Library Prep Kit. And finally, I would like to say that enzymatic fragmentation is highly tunable and special formulation reduces the sequencing bias. So let's move to the next topic, accelerating speed and performance of 16S workflow. So usually metagenomic studies are commonly performed by analyzing the prokaryotic 16S ribosomal RNA or 16S rRNA. And this A prokaryotic 16S rRNA is approximately 1500 base pair long, and it contains 9 variable regions. So typically, the variable region V3 and V4 of 16S rRNA are frequently used in phylogenetic characterization, such as to understand the genus and species in a diverse microbial population. So first, the 460 base pair region covering V3 and V4 is amplified using locus-specific primers. Then barcodes are added via another PCR step, and libraries are then sequenced in Illumina MySeq instrument, and subsequent data analysis reveals phylogenetic information of the microbial species. So what I would like to do here in next slide, I'd like to talk about the PCR steps with further details. So for the first step of PCR, it is done with locus-specific primers with some overhanged sequence. And then after first step of PCR, the PCR products get cleaned up using bead purification, and the PCR products from the first step used as an input for second round of PCR. For second round of PCR, Illumina Nextera adapters are used as primers. So during these PCR steps, you are introducing the index and P7, P5 sequences. Finally, the final round of bead cleanup, normalizing the samples, and libraries are pulled and eventually sequenced using MySeq V3 kit. Now, in this particular workflow, amplification or PCR is very, very important. And traditionally, scientists used Kappa Hi-Fi hot start ready mix. And what I'm going to show here in next few slides, that Spark Hi-Fi PCR master mix from QuantaBio is an equivalent but cheaper alternative of Kappa Hi-Fi hot start ready mix, and a better replacement will be Replica Hi-Fi top mix. So here is an example of tape station trace of the library prepared with Kappa Hi-Fi hot start ready mix. So it gives a clean single peak at 594 base pair. And similar libraries were prepared with Spark Hi-Fi PCR master mix. So if you compare these two libraries, the quality and yields of the libraries were very, very similar, as they really or nicely overlap with each other. In contrast, Replica Hi-Fi top mix generates similar quality of the libraries at shorter time. So if you look at how much time it took for the Kappa Hi-Fi or Spark Hi-Fi, it took a total of 75 plus 35. That means 110 minutes for 2 rounds of PCR. In contrast, Replica Hi-Fi top mix only took 35 plus 15, a total of 50 minutes for 2 rounds of PCR. So definitely Replica Hi-Fi is going to save a lot of time. And this was possible due to faster extension time of Replica Hi-Fi compared to Kappa Hi-Fi. So Replica Hi-Fi extension time is 1 second for this protocol compared to 30 seconds for Kappa Hi-Fi. And this will be extremely helpful for a high throughput lab where they're working on multiple different samples. Now, Replica Hi-Fi top mix is known to have high inhibitor tolerance. So we thought if it is possible to remove the bead cleanup step between first and second round of PCR. And that way, we wanted to see if we can save additional 30 minutes. And the idea was to take 2 microliter of the PCR product from the first round and directly used as an input for the second round of PCR. And if you look at the data, then you can see no difference between the situation where we did the bead cleanup and where we did not do the bead cleanup. So definitely we can remove this bead cleanup step and save additional 30 minutes. Now, we also try to combine 2 PCR steps together. So in a single PCR process, we introduce the locus specific primers and Illumina Nexter adapters and try to do both PCR steps at the same time in order to save the library preparation cost and some time. And as you can see here, you just need to set up one PCR reaction instead of 2. You can definitely save money and definitely save time because you do not need to set up another round of PCR. And we did that using Replica Hi-Fi Top Mix. And our results suggest that it is possible to combine 2 PCR steps together for a quick and dirty experiment. As you can see, the quality of the library was not as good as when you did 2 rounds of PCR. So you can still combine both PCR steps together if somebody is trying to do a quick and dirty kind of experiment. So from this section, we can say that Spark Hi-Fi PCR Master Mix is an equivalent replacement of Kappa Hi-Fi Hotstar Ready Mix in context of 16S metagenomic sequencing library preparation. And Replica Hi-Fi Top Mix produces equivalent library at faster speed from crude DNA extract. As I said, Replica Hi-Fi is a top mix. Even if you use, even if your input DNA is not so pure, it should be able to amplify from there. And using Replica Hi-Fi top mix, it is possible to combine 2 PCR steps together to save time and cost. Okay, so let's move to the last topic of the day, better structural variant detection in long-range sequencing. So in spite of monumental success and several advantages, traditional NGS with short reads, typically 150 to 300 base pair, poses few limitations. For example, it is limited in the ability to identify larger structural variants and mapping sequencing data into highly homologous and repetitive genomic regions and in case of phasing of alleles. And these limitations significantly impact the diagnosis of several genetic disorders. And sequencing with long reads can address some of these issues. There are few long-range NGS technologies available in the market. And in this presentation, we are going to focus on single molecule real-time or smart sequencing technology developed by PacBio. So smart sequencing has several advantages. For example, long reads of tens of kilobases can be achieved. So typically, PacBio sequencing handle between 500 base pair to 50 kilobase pair. And with their advanced data analysis method, sequencing results are highly accurate. You can get accurate data up to 99.99%. And uniform coverage throughout a wide range of GC content is possible. So how it works, similar to any other NGS method, smart sequencing has three steps. Uh, First of all, library preparation, sequencing using their SQL system, and eventually data analysis using SmartLink or custom pipelines. So for PacBio workflow, you can either starting from a double-stranded fragmented DNA or PCR product. So I just want to mention here that if you are fragmenting your human genomic DNA, for example, in order to get long reads, you are not fragmenting it into smaller fragments, right? So here you are making fragments of 20 to 50 kilobase pairs. And then SmartBell libraries can be generated from there. For sequencing, SmartBell libraries are annealed to sequencing primer and then bind to sequencing polymerase. Then this complex go through the sequencing process, generates multiple subreads, and during the data processing, these subreads are aligned with each other to generate consensus read called hi-fi reads. So you can use this long-range smart sequencing for multiple purposes. For example, whole genome sequencing, sequencing of RNA, sequencing of complex populations, targeted sequencing, and to find epigenetic signatures. In terms of today's presentation, we are going to focus on targeted sequencing using long-range PCR. So let's talk about targeted sequencing. So SmartBell libraries can be generated using high-quality long-range PCR products. So first step is generating long-range PCR. So I'll come back to that because that's the focus of QuantaBio. So once you have this long-range PCR product, then you need to ligate barcodes at both ends of your PCR products to generate SmartBell library. And for sequencing, as I mentioned earlier, it requires primer annealing, binding with sequencing polymerase, and finally generating consensus read for the libraries. So the quality of the long-range sequencing data vastly depends on the quality of the PCR amplicons. So if you have non-specific PCR products or incomplete amplification, that will often occupies sequencing space and will produce larger fractions of unusable data. And this in turn will impact the quality of variant calling by increasing noise in the sequencing data. And it will simultaneously increase the overall sequencing cost. So this problem can be addressed by using a high quality long range polymerase or long range master mix. So AccuStart long range supermix from QuantaBio is formulated to enable high quality, robust, reliable, and reproducible long range PCR. In our hand, this supermix was used to reliably amplify up to 24 kb of human genomic DNA and up to 40 kb of lambda DNA. And also the fidelity of this polymerase or master mix is 10 times higher than tag DNA polymerase. So because of this characteristic, your long range PCR products will be error free. So as you can see from this PCR setup table, because of this master mix, the PCR setup is very, very simple. It just required nucleus-free water, AccuStart long-range supermix, your primers, and template DNA. So setting up PCR is very, very simple. And if you look at the cycling condition, these are pretty standard. And I would like you to focus on the extension time recommended here. So AccuStar Long Range Supermix amplifies with fast extension speed, approximately 30 to 60 seconds per kb. And if you are amplifying a larger fragment, then definitely faster extension time like 30 seconds per kb will save you a lot of time. And I'll show you some data from our end how amplification worked with AccuStar Long Range Supermix. So in this picture, you can see that AccuStar Long Range Supermix demonstrates greater sensitivity and yield compared to other products in the market. And we were able to amplify a 8.5 kb region of beta globin gene using input DNA as low as 100 picogram. So if you have really small quantity of DNA, AccuStart long range supermix will be able to amplify your target gene. And here, using AccuStart long range supermix, we were able to amplify a 24 kb regions of TPA gene. And if you look at the quality of the band, it is a high quality, really distinct band, even at 24 kb fragment. So which is really important in order to get high quality sequencing data using long range PCR products. So we can definitely say that AccuStar Long Range Supermix will be a perfect candidate to be used in targeted packed bio workflow to generate long range PCR products. Finally, I want to show you this data set comparing Illumina data on the top and packed bio data in the bottom from the STRC gene. And you can clearly see from this data that long reads can easily identify multiple variants that was missed by short reads. So definitely hi-fi reads let users map and call variants that was missed by short reads. So finally, we can conclude that sequencing with long read can overcome challenges posed by short read sequencing. And AccuStart long range supermix generates high quality, robust, reliable, and reproducible long range PCR products for targeted sequencing. If you have any question or technical queries in future, please reach out to me via email. So here is my contact information. And if you have any product related information, please reach out to our global product manager, Monil Patel, via email. Or you can also reach out to us via our sales and customer service email. The other best way to reach out to us or get any product related information, so please contact your local VWR representative for any kind of query if you have any questions at this point.